So this is a safety and efficacy study of a novel compound, antibody drug conjugate called Tusamitamab ravtansine. This is an agent that is targeting CEACAM5, which is a protein on the surface of the cell, which is frequently expressed on non-squamous non-small cell lung cancer. And this monoclonal antibody has attached a cytotoxic drug called ravtansine, so that the monoclonal bind to the protein, the CEACAM5 protein in the surface of the cell. Then the whole antibody drug conjugate is interiorized into the cell. Then the linker that is joining the drug, the ravtansine, to the monoclonal antibody is, let's say, liberated intracellularly, and therefore the cytotoxic is free to kill the cell. Let's say that is the mechanism of action of the drug. And it is important because CEACAM5 is highly expressed in some 25% of those tumor cells. And therefore, having this specific payload, the antibody that conjugate is able to target those CEACAM5 expressing cells. The agent, the DM4 or ravtansine, is actually inducing immunogenic cell death, and therefore some studies in mice have shown synergistic anti-tumor activity when you combine the antibody drug conjugate to ravtansine with immunotherapy, with immune checkpoint inhibitors. So in this phase 2 study, the Carmen LCO5, we are actually evaluating Tusamitamab combination with pembrolizumab or with chemotherapy, with platinum, plus or less methotrexate plus pembrolizumab. So therefore, there are 3 cohorts into the study, one combining Tusamitamab with pembrolizumab, another Tusamitamab plus a platinum compound plus pembro, and then the fourth is a quadruplet, Tusamitamab plus platinum plus methotrexate plus pembro, and altogether we presented data on 25 patients. So as I mentioned, we have included 25 patients on those 3 cohorts. Important, in each cohort, some patients were treated at 150 milligrams of Tusamitamab and some others at 170. So altogether, in terms of efficacy, we have seen substantial efficacy. Half of the patients, 13 of the patients, had a partial response with an overall response rate of 52%, 13 out of 25 patients. Some additional 9 patients, 36% achieve a stable disease, only, let's say, for an overall disease control rate of 88%, and the median treatment duration is 24 weeks. Importantly, as I said, within the patients included in this trial, as you may imagine, some 2/3 of the patients had high expression of CEACAM5, and 36% had moderate expression. And I think that's an important thing to say. And importantly as well, some 88% of the patients had PD-L1 positivity in the tumor cells, being more than 50% in some 24% of the cases. Importantly, we have seen responses in all the cohorts treated with RAB plus pembro, with or without a platinum agent, with or without perimeter set. So at present, with this small number of patients, I don't think we can differentiate the activity we have seen with the quadruplet as with what we have seen with the doublet. Secondly, the responses were seen regardless, as I mentioned, the expression of PD-L1 and regardless the expression of SECAM5. At least this is what we have seen so far. And it's important to say that, the last follow-up, 7 patients had already duration of treatment of a year or more than a year, and still some 11 patients, 44% of the patients included, were still ongoing on treatment. Safety profile was, I would say, reasonable. Only we have seen on one case of dose-limiting toxicity, which was a transaminitis in a patient that received high doses of RAB, of Tusamitamab, 170 milligrams per square meter, and that was within the quadruplet. Most frequent adverse events treatment-related were nausea, 44%, diarrhea, 36%, and asthenia, 32%. Those were typically mild, and importantly, corneal side effects were observed in 24% of the cases. Only one case was keratitis grade 3. Most of the cases were grade 1 or 2. Importantly as well, pneumonitis were seen in some 16% of the patients, being grade 3 in 4%, and peripheral neuropathy occurred in 28% of the cases, being all of them grade 1 or grade 2. I think those data, the initial data, are promising of the combination of Tusarab plus pembrolizumab. It is encouraging to see that the preclinical data of synergy, potential synergy between this agent that induces immunogenic cell death plus pembrolizumab, a PD-1 inhibitor, are somehow confirmed in the initial patients treated here. That is giving some hope to maybe at some point translate this regimen into the first-line setting as a doublet, Tusarab plus Pembro, or in combination with other chemotherapy, platinum-based chemotherapy for patients particularly that are PD-L low expressors. I just have to say that we are, very thankful for patients participating in this trial and their caregivers, also the sponsor, for giving the support.